Alright, hello and welcome to another exam mini video. My name is Fisher Stines, and today I'm going to be talking about transcription in prokaryotic organisms using a train example. So to begin, I'd like to define a few terms here. So in our example, we can see that this is a strand of DNA, and we have our 5' end and our 3' end, as well as our 3' end and our 5' end. Now, for this specific example, we're going to be reading 3' to 5', and this is going to be the template strand. This is going to be our coding or non-template strand. Now, above this, we have something very special, and this is going to be our promoter region, our promoter station in this example, and it's going to tell the RNA polymerase, which is going to be represented by this train, what to do, and also, more importantly, where to bind. So it tells us about direction here, and tells it where to bind. Now say, for example, that we were to do a 5 base pair insertion between this -35 and this -10 region. Well, all of a sudden this promoter would look different, and this RNA polymerase would be unable to bind, rendering this ineffective. RNA transcription will not occur, the protein will not be made. So this is very important that this promoter region look correct. Now these numbers here correspond to something called a consensus sequence. And basically this is a very specific series of base pairs that tell the RNA where to bind. So a very common one here at the -10 region might be the Tata box, for example. So we have our promoter region, we have our RNA polymerase, and we're almost ready to get underway. And then we're going to add something called sigma factor. This gives us a little bit of chemical energy. We arrive here at the +1 point, and we are off to the races. So now what's going to happen is we are going to change up our train track ever so slightly and begin. So RNA polymerase is going to come into the place here. Our sigma factor is going to begin to disassociate, and with the help of topoisomerase, we're going to start reducing all of this torsional strain that exists in our DNA. It's going to open it up into this little transcription bubble, and it's going to get started. So the RNA polymerase is going to fly from 3' to 5', and we're going to start producing RNA in the 5' to 3' region. So we see we have a strand of RNA here, racing along the track, and then all of a sudden we have to come to a critical point where we're going to terminate. Now termination, we have two distinct possibilities. We have row independent, and this we're going to see a hairpin loop. So this is going to have guanine and cytosine sort of in rapid succession where they bind with one another to create this hairpin loop. It'll be followed by a series of adenines and uracils. This will dissociate. The secondary option is going to be use something called a row-dependent termination, and this is used as this row protein. It's going to attach at the 5' region. It's going to sort of chase up to the 3' region, and it acts very similar to like a helicase or a topoisomerase. It's going to disassociate, unwind these two RNA and DNA strands, and boom, we lose our RNA strand. So thank you very much. This has been my exam mini video.